bacterial single staining, and using microscope, microbiology laboratory skills. Part 1, Bacterial Single Staining Introduction. Because most microorganisms appear almost colorless when viewed through a standard light microscope, we often must prepare them for observation. One way to do this is to stain, color, the specimen. Staining simply means coloring the microorganisms with a dye that emphasizes certain structures. Before the microorganisms can be stained, however, they must be fixed, attached, to the microscope slide. Fixing simultaneously kills the microorganisms and fixes them to the slide. It also preserves various parts of microbes in their natural state with only minimal distortion. Stain is applied and then washed off with water, then the slide is blotted with absorbent paper. Without fixing, the stain might wash the microbes off the slide. The stained microorganisms are now ready for microscopic examination. A simple stain is an aqueous or alcohol solution of a single basic dye. Although different dyes bind specifically to different parts of cells, the primary purpose of a simple stain is to highlight the entire microorganisms so that cellular shapes and Bocce structures are visible. The stain is applied to the fixed smear for a certain length of time and then washed off, and the slide is dried and examined. Simple stain, only one stain is used, and all types of bacteria appear as the color of that stain when viewed under the microscope. Simple stains can be used to determine a bacterial species' morphology and arrangement, but they do not give any additional information. Occasionally, a chemical is added to the solution to intensify the stain. Such an additive is called a mordant. One function of a mordant is to increase the affinity of a stain for a biological specimen. Another is to coat a structure, such as a flagellum, to make it thicker and easier to see after it is stained with a dye. Some stains commonly used for simple staining in the laboratory are methylene blue, carbofluxin, crystal violet, and safranine. Be sure to fill the entire smear with the dye. Leave the stain on for 30 seconds. Gently wash the slides with distilled water. Drain off any excess water. Use blotting paper to gently blot the slide dry. After the slide is completely air-dried, the smear will look like a thin colored haze. And your smear ready to observe under the microscope. Part 2, Observing microorganism through a microscope. Microorganisms are much too small to be seen with the unaided eye. They must be observed with a microscope. The word microscope is derived from the Latin word micro, which means small, and the Greek word scopos, to look at. Microbes must undergo several staining procedures before their cell walls, capsule, and other structures lose their colorless natural state. The best way to observe bacteria is by using a compound light microscope. This type of microscope provides the necessary magnification and resolution to visualize bacteria. To ensure accurate observations, it is important to follow proper techniques. Unit of measurement. Microorganisms and their structural components are measured in even smaller units, such as micrometers and nanometers. A micrometer, μm, is equal to 0.000001 m, 10 to 6 m. The prefix micro indicates that the unit following it should be divided by 1 million, or 106. A nanometer, nm, is equal to 0.00000001 m, 10 to 9 m. Angstorm, A, was previously used for 10 to 10 m, or 0.1 nanometers. This figure shows various specimens that can be resolved by the human eye, light microscope, and electron microscope. Light microscope. A light microscope is an optical instrument used to view objects too small to with the naked eye. It is so called because it employs the use of white or visible light to illuminate the object of interest so it can be magnified and viewed through one or a series of lenses. A light microscope, also known as an optical microscope. The basic principle behind a light microscope involves passing light through a sample and capturing the resulting image. Light microscopes can produce a wide range of magnifications, typically ranging from 40x to 1000x, or even higher with specialized objectives. Parts of the light microscope. These are the primary components of a light microscope, though additional features and accessories may vary depending on the specific model and intended applications. The combination of these parts allows researchers and students to explore the microscopic world and study various specimens with enhanced magnification and clarity. Eyepiece lens or ocular. Lenses that the viewer looks through, the lens closest to the observer's eye. The eyepiece, or ocular lens, further magnifies the image and allows the observer to view it. By combining the magnification of the objective lens and the eyepiece, the overall magnification of the image is determined. Body tube connects the eyepiece to the objective lenses. The body tube contains a series of lenses that further magnify and transmit the image from the objective to the eyepiece. In some microscopes, the body tube can be extended or tilted for user comfort and ergonomic positioning. The objective lens are a set of lenses positioned close to the specimen. They gather and magnify the light that passes through the sample, creating an enlarged image. Microscopes usually have multiple objective lenses with different magnification powers, such as 4x, 10x, 40x, and 100x. They are typically mounted on a rotating turret, allowing the user to switch between objectives easily. Stage. The flat platform where the slide is put in place for viewing can be adjusted via the coarse and fine adjustment knobs. The stage may include clips or mechanical stages to secure the specimen in place, called stage clips, which used to keep the slide in place. The stage can be moved vertically or horizontally to position the specimen for examination. Condenser lens is located below the stage and plays a crucial role in focusing the light onto the specimen. It consists of a series of lenses that concentrate and align the light source to provide uniform illumination of the specimen. The condenser can often be adjusted in height or aperture size to control the intensity and angle of the light. Iris diaphragm is an adjustable apparatus located below the stage, within the condenser, which can be manually modified to vary the intensity of light that reaches the specimen so the user can regulate the intensity and contrast of the image. Illuminator is the source provides the light that passes through the specimen. It is typically located in the base of the microscope and may consist of a halogen lamp, LED, or other light sources. The illumination can be transmitted through the sample, transmitted light microscopy, or directed onto the sample from above, reflected light microscopy. Base. The flat structure of the microscope that supports the entire instrument. It often contains the illumination source, power switch, and electrical controls. The base helps maintain the stability of the microscope during usage. Arm connects the base of the microscope to the nosepiece and eyepiece, used to carry the microscope around. The arm is the curved or angled portion of the microscope. Eyepiece adjustment knobs used to bring the specimen into focus. Light microscopes have two focus knobs, one for coarse adjustment and the other for fine adjustment. These knobs move the stage or objective lenses vertically to bring the specimen into sharp focus. The coarse adjustment knob is used initially to bring the specimen into view, while the fine adjustment knob provides precise focusing. Steps on how to use a light microscope. Connect the light microscope to a power source. The light source, typically a halogen lamp or an LED, emits light that passes through the condenser lens. The condenser lens focuses and directs the light onto the sample, illuminating it. The light then interacts with the sample, undergoing various interactions such as absorption, scattering, and reflection, depending on the nature of the specimen. If your microscope uses a mirror instead of an illuminator, you can adjust the mirror, illuminator, and/or diaphragm for maximum light exposure. Instead, find a place where natural light is easily accessible. Turn the revolving nosepiece so the lowest objective lens is in position. Placing a drop of immersion oil between the microscope objective and the microscope slide. To archive high magnification 1000x with good resolution, the objective lens must be small. Although we want light traveling through the specimen and medium to refract differently, we do not want to lose light rays after they have passed through the stained specimen. To preserve the direction of light rays at the highest magnification, immersion oil is placed between the glass slide and the oil immersion objective lens. The immersion oil has the same refractive index as glass, so the oil becomes part of the optics of the glass of the microscope. While oil immersion is not always necessary to see bacteria, it is often employed for higher magnification observation and enhanced clarity. The use of oil immersion microscopy is particularly beneficial when studying smaller bacteria and finer bacterial structures. The primary advantage of oil immersion is its ability to increase the numerical aperture, NA, of the objective lens, which determines the resolving power of the microscope. Higher numerical aperture allows for better resolution, enabling the observation of smaller details within bacterial cells. Without oil immersion, the quality of the image may degrade due to refractive index mismatch between the glass slide, the specimen, and the objective lens, resulting in sharper and clearer visualization of bacterial cells and their intricate details. However, it's important to note that oil immersion is not required when using lower magnification objectives, example, 10x, 20x, or when studying larger bacteria. In such cases, a cover slip can be used to maintain a thin layer of liquid, typically water or a suitable bacterial growth medium, between the specimen and the objective. Mount your specimen onto the stage. Use the metal clips to keep your slide in place. Make sure the specimen is positioned in the center, right under the objective lens. Gently move the 100x objective into the oil, without disturbing the slide. Look through the microscope. You should see the stained material, but it will probably out of focus. Look into the eyepiece and slowly rotate the coarse adjustment knob to bring your specimen to focus. See to it that the slide does not touch the lens. Now slowly rotate the fine adjustment knob until you obtain a clearer image of your specimen. You may have to move the slide slightly if the stained cells are at the edge of the field. Once you have the slide in focus, you can move to other fields to see more of the bacteria. Examine your specimen. Always keep both eyes open. Even though you're only using one eye to look through the lens, closing the other eye can strain your eyes. And remember, everything is backward and upside down. Moving the slide to the right puts the image to the left and vice versa. During examination, attention should be given to the bacteria's shape, arrangement, and any other observable features. Detailed notes, sketches, or photographs can be taken to document the findings accurately. Maintaining a sterile environment throughout the process is crucial to prevent contamination and obtain reliable observations. Following the specific instructions provided with the microscope and adhering to proper laboratory practices ensure optimal results. Overall, using a compound light microscope, preparing the slide properly, and employing staining techniques when necessary provide the best way to observe bacteria and study their characteristics effectively. What is the necessary magnification to see bacteria with a microscope? To view bacteria under a microscope, various magnifications are typically used. The specific magnification depends on the size and level of detail required for observation. The most commonly used magnifications for viewing bacteria are low magnification, 10x to 20x. This magnification is useful for locating and surveying the bacterial sample on the microscope slide. It provides an initial overview of the distribution and arrangement of the bacteria. Medium magnification, 40x to 100x. This range of magnification allows for more detailed observation of bacterial morphology, such as shape, size, and cellular structures. It provides clearer visualization of individual bacteria and their characteristics. High magnification, oil immersion, typically 100x to 1000x. High magnification objectives are used for finer examination of bacteria. Oil immersion microscopy is often employed at this level to maximize resolution. It involves placing a drop of immersion oil between the slide and the microscope objective to reduce light refraction and improve image clarity. This magnification is crucial for studying smaller bacterial structures, such as cell walls, flagella, and pili. It's important to note that the maximum useful magnification of a compound light microscope is typically around 1000x. Beyond this, the resolution becomes limited by the wavelength of visible light, and electron microscopy is usually required for higher magnifications. Choosing the appropriate magnification depends on the specific objectives of the study and the characteristics of the bacteria being observed. It may involve using different magnifications sequentially to obtain a comprehensive understanding of the bacterial sample. Is it possible to magnify a microscope to that level that bacteria become invisible? A microscope can be zoomed in too far to see bacteria clearly. There is a limit to the useful magnification of a light microscope, determined by the resolving power of the microscope and the wavelength of visible light. When the magnification exceeds this limit, the image may become blurred or distorted, making it difficult to see fine details such as bacteria. The maximum useful magnification of a compound light microscope is typically around 1000x. Beyond this point, the resolution is limited by the diffraction of light, and the image quality deteriorates. Increasing magnification without improving the resolving power of the microscope does not provide clearer or more detailed views of bacteria. To overcome this limitation and observe bacteria at higher magnifications, electron microscopy techniques are employed. Electron microscopes use a beam of electrons instead of light, allowing for much higher magnifications and resolutions. Transmission electron microscopy (TEM) and scanning electron microscopy (SEM) are commonly used to visualize bacteria and other microscopic structures at extremely high magnifications. Therefore, when using a light microscope, it is important to select an appropriate magnification level within its range that allows for clear observation of bacteria while considering the resolving power of the microscope and the size of the bacteria you are studying. The true maintenance and cleaning of a microscope the right way. A good quality microscope is not cheap. Proper care and maintenance ensures your device performs at its best. Here are important tips on how to handle your light microscope. Never hold the microscope by the piece. Support the stand and hold the arm when carrying the instrument around. Always carry a microscope upright, as the eyepiece could fall off. Always turn the illuminator off after you use it. Use a non-solvent cleaning solution to avoid damaging the lenses. Use a microfiber cloth when wiping off dust and dirt from lenses. There are microscope cleaning kits you can purchase to make microscope cleaning safer and more adequate. When the microscope is not in use, cover it with a dust jacket. While using the microscope, do not rush through the viewing process. Be careful when handling the knobs, and avoid turning the nosepiece unnecessarily, as they can wear out.